After learning about the anatomy and the physiology of the respiratory system, here comes a big question, an important question. How is breathing regulated? Well, if we know the reflex arc, where a sensory input would be taken to the control center, where it's processed and a response is given via a motor nerve to the target structure, which would then respond to the signal. So it's a very similar mechanism that exists in the respiratory system. We are talking about chemoreceptors, we are talking about mechanoreceptors. All right, mechanoreceptors could be in the lungs, airways, muscles, joints, and there could be other signals like pain, pressure from the cardiovascular system. These things all reach the central nervous system. In the central nervous system, a specific part of the brain that's directly involved in the regulation of breathing is the brainstem. As you know, the brainstem consists of the midbrain, the pons, and the medulla. We have both pons and medulla play a significant role in the regulation of breathing. There are two centers in the pons, there are two centers in the medulla. The centers in the medulla are the ones that directly are involved, which are influenced or regulated by the centers in the pons. However, there are other centers like the hypothalamus, like the cerebral cortex. They have inputs from there to the brainstem. All right, fear, anxiety, and things of that sort could be emotional, and emotional center happens to be part of the hypothalamus. So hypothalamus can send signals to the medullary centers, and any voluntary regulation that is required, such as when you're singing and acting, all these, purposeful breathing changes, voluntary changes could be due to the signals arriving from the cerebral cortex to the medulla. And then what does medulla do? Send signals through these efferent neurons, motor neurons, to the muscles of breathing. We already know these muscles, mainly the diaphragm, and then maybe the external intercostal muscles. So they control the muscle contraction. So muscle contraction that's responsible for ventilation, that's inspiration, is not by the decision of these muscles by themselves, but by the instructions sent by the brain centers, such as the medullary centers. Medullary centers, as you know, do not work independently. Important for us to know. They are in turn controlled by many more centers. All right, so this is just an overview of how breathing is regulated. Now we will go into additional details. As we learned, we have respiratory centers, many of these, two in the pons, two in the medulla, which are yet again controlled by the higher centers. The afferent and efferent nerves, afferent are the sensory nerves, efferent are the motor nerves. Okay, we could talk about the afferent nerves that control breathing. All right, vagus nerve that supplies the lungs could be taking any stretch, any pressure within the lung to the brainstem. That's just one example. All right. And again, there are many other structures we talked about, like from the skin, from the joints, from the muscles, and so on. So they are coming from many different sensory neurons. All right. Efferent nerves that originate in the CNS, that is at the medullary level or at the cerebral level, they then ultimately would reach via the spinal cord typically, right? And then from the spinal cord to the inspiratory muscles. Diaphragm is controlled by phrenic nerve. Phrenic nerve is made of C3, C4, C5. These are cervical spinal nerves that come together to form the phrenic nerve. Anytime any neck injury, automobile accident, neck injury, sudden death could be correlated to the damage to phrenic nerve. Phrenic nerve damage directly affects the function of the diaphragm. So if diaphragm cannot move, then suffocation and ultimately death. In any case, that is how diaphragm is controlled. External intercostal muscles are controlled by the intercostal nerves. Those are the spinal, thoracic spinal nerves, T1 through T11. All right, so those are the motor or the efferent nerves reaching the respiratory muscles, but there are many other neurons between the cerebral cortex and the brainstem, between the hypothalamus and the brainstem, and so on. Sensory receptors, as I mentioned, mechanoreceptors are involved, baroreceptors are involved. By the way, baroreceptors are a type of mechanoreceptors. Chemoreceptors are involved. All right, pain receptors, and so on. All right, so now this is the illustration of the location of the major respiratory centers. This is our medulla here. This is the pons. There are two centers in the pons, pneumotaxic center and then the apneustic center. Okay, both together are called pontine respiratory group. Pontine respiratory group, as you can tell, reach the medulla, and in particular the dorsal respiratory group. Dorsal respiratory group is like the primary center that stimulates the respiratory muscles. So this is an inherent the natural stimulatory signal originating in the dorsal respiratory center, DRG, dorsal respiratory group. Ventral respiratory group typically is not as involved in quiet breathing. All right. So it may regulate the rhythm to some extent, but then not as important as our dorsal respiratory group is. I also want you to pay attention to the signs here, plus and the minus. Very important, very important. If you look at the pneumotaxic center, it has inhibitory signals. Inhibitory signals because this is the one that limits the contraction of the muscle from the dorsal respiratory group. Just imagine your muscles in a contracted state the entire time. That would be impossible, isn't it? You have to let these muscles relax. And that kind of signal originates from the pneumotaxic center. Again, although you see that as inhibitory, this inhibition is critical in turning off the inspiration and initiating expiration. So that is the job of pneumotaxic center. Apneustic center simply stimulates the function of dorsal respiratory group. In other words, echoes or supports the stimulatory function that the dorsal respiratory group does. All right. Now, what you're looking at is the intercostal muscles here and the diaphragm, the internal intercostal muscles here, and then the accessory respiratory muscles here. Diaphragm would be right here. This is the one that is supplied by phrenic nerve. This is the one by the intercostal nerves. There are other nerves originating internal intercostal muscle, again from the thoracic spinal nerves, and then accessory respiratory muscles could be again from the thoracic and basically T1 to L1 supply the abdominal muscles, and C1 to C2 those are accessory muscles that supply the neck. All right, so these are efferent nerves supplying the various respiratory muscles. With this information, let's talk about each and every group that makes up the respiratory center. The main one, as you know, is the dorsal respiratory group. This has this intrinsic nerve activity to stimulate the respiratory muscles. So this acts like a pacemaker of breathing. All right. The way it acts is also interesting. It would start off in a weak manner. So you're looking at this weak raise, and then the response would increase steadily in a ramp manner for about 2 seconds. So 2 seconds here of inspiration, put it that way, and then ceases abruptly. This is probably where you think about the action of pneumotaxic center setting in, right? That's the one that turns off that stimulatory signal from the dorsal respiratory group. So there is that certain change in pattern. So now this is going to be the expiration part. So this little L here would be our expiration. So this last 3 seconds, this last 2 seconds, all in all, it would be 5 seconds for one breath. Okay, typically 12 breaths per minute. That's because of the way each breath takes 5 seconds, approximately. All right, so now let's talk about pneumotaxic centers. This is the second most important one. Remember, dorsal respiratory group is the most direct one, turning on the contraction of the respiratory muscles, whereas pneumotaxic center is the one that gives a break between inspiration and then initiates expiration, correct? So limit inspiration, that ramp effect is now stopped. Correct? So now the inspiratory area, dorsal respiratory group, is the one that's controlled by pneumotaxic center. And apneustic center is the one that triggers the function of the dorsal respiratory group. So it increases the respiratory rate. So more frequently, pneumotaxic center is activated, more frequent the break is, so the breathing cycles are more per minute than in a resting situation. All right, so control the rate of respiration by controlling the switch off point of inspiratory ramp, control the duration of the filling phase of the lung cycle. So a strong signal from the pneumotaxic center Now you are only inspiring for 0.5 seconds instead of 2 seconds before. It's now only 0.5 seconds, and therefore you're breathing faster, filling less in each breath. On the other hand, if the signals are weak, and if pneumotaxic center does not turn off the inspiratory ramp, the inspiration can go on for as long as 5 seconds, and that would help fill more air. So slow breathing here and fast breathing here is what you're looking at, and all of that is determined by the activation of the pneumotaxic center. As I mentioned before, apneustic center increases depth of respiration by acting on the inspiratory center. Ventral respiratory group, again, not as active during quiet breathing, and it would have overdrive mechanism when more of the ventilation is needed. Okay, provide expiratory signals to the abdominal muscles during heavy expiration. Well, we already talked about the afferent and efferent pathways. This is a good time for us to revisit this. You're talking about some cranial nerves here. Cranial nerve 9 are glossopharyngeal, and cranial nerve 10 are vagus. They are the ones that bring signals from the chemoreceptors and baroreceptors to the brain stem area. Receive signals from the thermoreceptors, pain receptors, and proprioceptors as well. So there are so many other nerves that are not listed here, but just know that this could be from the skin, from any other part of the body that is reaching the brainstem with the signals. The efferent pathway is the motor pathway that originates at the brainstem and reach the muscles of respiration via the spinal cord. All right, motor signals through anterior portion of the lateral column of the spinal cord. Cervical and thoracic segments of the spinal cord are more involved. We talked about phrenic nerve made of C3 and to C5, supplying the diaphragm, in external intercostal muscles T1 through T11, supplying intercostal muscles. Now we are switching from the neural regulation to chemoregulation. This is basically chemical substances in the body fluids and how they could affect breathing. All right, so now the chemical substances that we mainly discuss here are the levels of oxygen, hydrogen, CO2. Okay, hydrogen ion concentration is directly a measure of pH, as you know, right? So if you have too much hydrogen ion in the blood, that would be low pH, and vice versa. All right. So if there is slow breathing, there may be a buildup of CO2. So high levels of CO2 can trigger chemoreceptors. And if for some reason the lungs are not bringing enough oxygen, again, could be due to slow breathing, decreased levels of oxygen could be another change that would trigger chemoreceptors. All right, so mainly we talk about oxygen, CO2, and hydrogen ion concentration. And then hypoxia is low levels of oxygen, and hypercapnia is too much CO2, and we talked about hydrogen ion concentration. So two types of chemoreceptors. Those are central chemoreceptors and peripheral chemoreceptors. Central chemoreceptors are within the central nervous system. Peripheral chemoreceptors in the peripheral nervous system. Very easy to define those. All right, so here is a summary of which ones are detected by which type of receptors. High levels of CO2 is the main trigger for the central chemoreceptors. And high levels of hydrogen ion concentration. By the way, high level of CO2 can affect the high levels of hydrogen ion. That they're interrelated. Both could trigger central chemoreceptors. Peripheral chemoreceptors are very, very sensitive to low levels of oxygen. They are also sensitive to high levels of CO2 and hydrogen ion. Please note that. However, they're more sensitive to low levels of oxygen than the high levels of CO2 and hydrogen ion. That's how you have to interpret that. So anytime there is hypoventilation that leads to hypoxia and then hypercapnia, what would happen? You have the activation of peripheral or central chemoreceptors. All right, so here is an example. High levels of CO2 and high levels of hydrogen ion would trigger the respiratory center, and that in turn would trigger inspiration. So anytime there is a buildup of CO2, there is a buildup of hydrogen ion that would trigger the respiratory center to initiate faster breathing. All right, you call it as hyperventilation. Hyperventilation would help get rid of the CO2 that was in a higher concentration and return things back to normal. So it's a great example of homeostasis. All right, chemosensitive area. Well, again, the area within the brain that is sensitive to levels of CO2 and hydrogen is one particular area, again in the brainstem, very close to the medullary breathing centers. All right. So now what do they do? They are sensitive to high levels of hydrogen ion in the cerebrospinal fluid. And here is a great depiction of what happens if there is hypoventilation, slower breathing. All right. There is a buildup of CO2. There is drop in pH. That's because, too much CO2 triggers the buildup of hydrogen ions, and then that leads to low pH. This is detected by chemoreceptors. These are peripheral chemoreceptors that they're talking about. Peripheral chemoreceptors, by the way, are in the aortic arch and in the carotid bodies. These are the same locations where you have baroreceptors as well. Okay. In any case, they're activated. They take the signal to the medullary respiratory center, and then what do you expect? The breathing is now stimulated, faster breathing than normal, and so that is through the signals traveling via spinal cord to the respiratory muscles and increased ventilation. So decreased ventilation ultimately leads to increased ventilation. This is a nice example of negative feedback, correcting the situation. What we're also looking at is if we have high levels of carbon dioxide, and that is detected by the chemoreceptors within the medulla, and they also trigger the respiratory center. So you're looking at two mechanisms being turned on simultaneously, the central chemoreceptors, peripheral chemoreceptors. All of these simply increase the rate of breathing in order to get rid of this excess CO2 and bring the situation back to normal. And this is where you see the chemosensitive area, and this is supposed to be the the medulla, where you would see the sensory area. And what it's trying to show you here is that if you have a buildup of CO2, all right, you know how CO2 can combine with water and then dissociate to become hydrogen ion and bicarbonate ion. Okay, that reaction occurs to some extent in the blood. And hydrogen ions are not allowed to cross the blood-brain barrier. So that's what it says. So hydrogen ions do not reach the brain from the blood. However, CO2 can. CO2 is quick at reaching the brain. High levels of CO2 sensed by the central chemoreceptors, and they start making the respiratory breathing faster. All right, so here you're talking about high CO2 and high hydrogen ion concentration from its conversion to hydrogen ion. In any case, there is decreased pH, you see. Now, so what happens is you're triggering the breathing, but then bicarbonate ion eventually would reach the brain through the blood-brain barrier. So it says here slow, its crossing of the blood-brain barrier is slower than that of CO2. So what happens, therefore, is that initially you have triggered the breathing rate, but then you notice that bicarbonate will control the buildup of hydrogen ion, and therefore things should plateau and then correct themselves. All right, so initial change would get rid of excess CO2 and things should be returning back to normal. This is just to show you the location of the peripheral chemoreceptors, and very sensitive to decreased PO2. As I mentioned, peripheral chemoreceptors are very sensitive to decreased PO2, more so than decreased the buildup of too much carbon dioxide or hydrogen ion. And that's the end of the respiratory anatomy and physiology series of lectures. Thank you very much.